So hello, and today I'm going to talk about this paper called Visual Object Categorization in Infancy by Spirit and colleagues. So I've chosen this paper for a couple of reasons. The first one is that it is relevant to a potential PhD direction that I want to take, which is on the similarity score on visual objects. And on the other hand, the paper has also tried to tie the phenomenology of, in this case, infants' object qualia, so to speak, to some neuroscientific measures like fMRI. And the connection from the phenomenology to the neuroscientific measures is also something that I want to take on in my PhD as well. And since now has talked about going a little bit outside the comfort zone, so here I'm looking at infants study, which I'm not, to be honest, I've this is the first paper that I've read about infants psychology. And also, I know very little about this type of similarity rating in non-adult performed in non-adults. So therefore, it's something that's what interests me when I have a look at the paper. So the paper, as we mentioned, is looking at visual object categorization in infants. So the way in which the paper is conducted is they recruit infants that are at 4 months, 10 months, and 19 months old. And then for these infants, they are asked to look at basically images of objects. So these are shown on the top right corner. There are a total of 36 images shown in 8 categories. So they are broadly categorized into animate versus inanimate objects. And then within them, you've got human versus non-humans, and then natural objects versus artificial objects. And what they have done, what the researchers have done, is to ask the infants, or they show the infants, 2 items or 2 of the 36 images, and then record the looking time at each of the objects using eye tracking. And then what they've done, well, there are a lot of analyses that I've done, but the main one that's most relevant to qualia structure is this difference in looking time, DLT. So this is calculated on a pair of the basis of a pair of objects. So if you have 2 objects, then DLT is calculated as the difference in looking time divided by the sum of the looking time, right? And in this case, then DLT could be calculated per object pair, and then they can be compiled into a matrix to turn into a so-called dissimilarity matrix for infants. And then what they've done is then use this dissimilarity matrix to compare with hypothetical models or fMRI-based models that uses the same set of images. I'm going to talk a little bit more about the models as we go through later on. Okay. So there are 2 basic directions that the paper has taken. These are not necessarily experimental. These are not these are not experiment and experiment 2 in the paper. I've only looked at experiment 1, just because of the time, and also within experiment 1, there seems to be 2 directions. The first one is to compare infants categorization to adults categorization. And in this case, there are basically 2 ways that the authors have tried to do that, to ask this question. The first is to compare the dissimilarity matrix in infants with these hypothetical models that they presume adults would have. And we're going to talk about the hypothetical models in the next slide. And if the dissimilarity matrix align with these hypothetical models, then they conclude that the infants can categorize on this one particular criteria. On the other hand, they have also looked at between and within category difference in looking time. So what it means is that for a single category, say human face, if the infants look at 2 human faces for a similar amount of time, then the within category DLT would be small. However, if it look at objects from another category, say non-human faces, significantly longer or shorter than looking at human faces, then the between category DLT is going to be big. And using this comparison of within to between category DLT, then if the between category DLT is significantly bigger than the within, then they conclude that the infants can distinguish the category of objects from others. So what they have realized is that these two methods basically gave them the same conclusion. So therefore, in the next slide, when I explain the results, these will be combined together. So let's talk a little bit about the hypothetical models. So we've mentioned that there are 8 categories of objects, and these are the models on the right that they have generated. So for each of the category, they hypothesize that some certain subject can distinguish between based on these criteria perfectly. So for example, in the 8 category model, they presume that this subject would perfectly distinguish between human faces, between all 8 categories, so that only those that are those that have the same category would be identical, and those that are not in the same category would be maximally different. So on the other hand, with say animacy, then the model presumes that they can only distinguish between animate versus inanimate objects, so that each category of animate objects, which we have 4 of them, are identical to one another, and each inanimate objects are also identical to one another, but those that are inanimate versus animate would be maximally different. So therefore, they are in blue, so to speak. Hopefully that makes sense as to how they make the model out. We may talk a little bit about more about it in the Q&A, because I find it to be quite confusing to grasp at the start as well. Okay. So anyhow, by comparing the inference dissimilarity matrix with the hypothetical ones, and as well as looking at the between versus between category difference in looking time, they found that 19-month-old infants can categorize based on animacy, that is, whether 2 objects are animate versus inanimate, humanness, that is, whether a particular object belongs to a human or a non-human animals, and they can basically distinguish between the 8 underlying categories. And then on the other hand, for the 10 months old, their categorization is not as good as the 19 months. They can only categorize based on animacy, that is, they can distinguish between animate versus inanimate objects. And then for 4 months old, they can't even distinguish based on animacy, and they can only categorize based on low-level visual features like the size of the image, the elongation, and then compactness of the image. So, so that was the result for the first part. The second part, which I find to be even more confusing than the first, is where they try to compare infants' pattern of object categorization to patterns of activity in the adult visual cortex. So in this case, what they've tried to do is to compare the dissimilarity matrix of the infants with the dissimilarity matrix about fMRI activation in a certain area when an adult is looking at some object images. So in this case, they have looked at 3 areas in the fMRI. So for these, you would call this dissimilarity matrix. They are looking, each pixel represents the difference in fMRI activity at a certain region when they're looking at image 1 versus image 2. And in this case, they have compiled 3 dissimilarity matrix to compare from adult early visual cortex, adult ventral occipital temporal cortex, and adult lateral occipital temporal cortex. And what they have found was that for 19 months old, their pattern of recognition, that's their dissimilarity matrix, aligns quite nicely with the fMRI dissimilarity matrix in all 3 regions, and also the majority of the visual ventral stream, which is used to process objects. And then for the 10 months old, they realized that their dissimilarity matrix in 10 months old infants does not align with these regions that we've mentioned, but align with some parts of the visual ventral stream. And finally, for the 4 months old, their kind of categorization is not similar to any part of the adult visual cortex. So I find this to be quite confusing, but anyways, we'll move on to the conclusion. So the conclusion was that for 4 months old, infants can look at, well, they can look at certain visual aspect of the image, so they can distinguish between large and small images. And what they've realized is that since most of the images, most of the human faces images are quite large in their sample size or in their image set, therefore infants can categorize based on faces compared to, say, bodies. Then for 10 months old, they can distinguish between animate and inanimate objects, as we've mentioned. And then for 19 months old, they can distinguish into a bit more complex categories, say inanimate or artificial versus natural objects or not humans. And suddenly, I thought that was quite odd when they say that as infants grow older, their behavior correlates with the neural activity in larger aspects of the adult cortex. I find it a bit confusing as to how this conclusion makes sense, to be honest, and that also leads me to the things that I don't understand. The first part is about the comparison between adult fMRI results and the phenomenological data from infants. What they're trying to do is to compare, yeah, basically fMRI data with phenomenological data. But in my opinion, I thought these two are categorically different stuff or different data, and therefore they cannot be comparing. They're basically comparing apple with oranges, which I think is a little bit odd, and I'm not quite sure how to interpret that. And on the other hand, they have proposed that difference in looking time to be a measure of dissimilarity rating, so to speak. The author says that this is the first time that anyone has done this, and I'm not sure if there are other ways to measure dissimilarity rating in non-adult subjects, say in this case in infants, or maybe in other animals as well. And yeah, and also there's a question in the back, is this DLT a good proxy for dissimilarity as well? And finally, in experiment one, what the authors have done is to compare the empirical data with some hypothetical models. So they are trying to say that infants can categorize some categories if their data aligns with the hypothetical model. And I'm not sure, and perhaps Li Peng could comment on this because you're quite good at data statistics in general, and to see that if there are ways in which you can extract this type of information that they are able to categorize between various categories using some other analysis method rather than comparing with hypothetical models. Because I'm not quite familiar, yeah, this is the first paper that I've seen that they have used a hypothetical model. So I'm quite generally quite new to the literature in general, so I'm not sure if this is a standard practice or it is something that's a bit odd. All right. And finally, the things that I wish to discuss in the grant meeting a little bit more. The first is about comparing neural data with phenomenological data. So this is one of the things that the paper has tried to do is to compare fMRI results with, in this case, difference in looking time results. I don't think they have done it quite convincingly, but I'm not sure if there are ways that that others have looked into it and see how that should work. And also, I'm interested in animals and neuroscience in general because that's my undergrad, right? So yeah, I'm interested in any works with animals or anything that is looking in, say, neurotransmitters, or neuronal activities and so on. And finally, I'm also interested in any work that is working on color qualia because my intern project at the moment with now is on color and color blindness in general. Work on color will be interesting to me as well. And yeah, I think I'm quite on time actually, just about 7 seconds off. And yeah, that's my presentation. Thank you.
Thank you.
Thank you.
Should we start with the Q&A? Anyone wanna kickstart? Yeah, the comments.
Actually, I have the same question as you, the previous slide, maybe. Yeah, the first one and second one are the same. I have same question. And yeah, I also think that they should compare the DLT from adult
mm,
and fMRI data, and then they uh, compare the infants data and the adults fMRI data. But yeah, did did they do the uh mm, yeah, they didn't measure the looking time from adult, right?
I don't think so. And even for the fMRI, they did a whole experiment on fMRI to get the fMRI matrix, but they have sort
Mm,
of just in the paper they have sort of shown it as if it's just a given. Like this is the matrix that we've got without talking too much about how they collect that data out. It's in the method, but it seems to not be the main focus of the paper. Yeah.
mm,
Okay, so okay, I think I
Yeah.
have some idea regarding these two questions. I will take the second question first because
mm,
I think I'm a psychology student, so this is quite familiar in development psychology, the course that I learned. So looking time
mm,
or gaze time in infant investigating this thing this time is very usual in development psychology because they assume that when infant seeing a new stimulant they have never seen before, they are very curious. So they will spend more time in watching that stimuli. But if they have seen this stimuli many times, so they will get bored. So they will move their eye to the other stimuli. That is called habituation and dishabituation. So in this way, the scientists can identify whether infant can discriminate new stimuli and then old stimuli. So in this case, two same thing, for example, two faces, they are from same category, right? So in certain extent, they are same thing. But if we present one pen and then or what an animal and then a human face, they are actually two different things. So the infant will spend maybe different time on two different category. So I think that's what they want to uh why they use this looking time as a proxy of this similarity.
But I think that would introduce a problem, right? Because as you mentioned,
Mm,
the infants are gonna look at novel objects more than familiar objects. That's as you mentioned is probably quite established or quite commonly used in developmental psychology. So if you show, say, if the infants, I don't know, just it's more fun. So if you show them a toy that they play a lot,
mm,
and then on the other side you show them a toy that is not gonna be that they have never played before, then they're gonna look at the second one a little longer, right, or a lot longer. But then, but then that is not really just categorization. That is just recency effect, or there's just familiarity effect rather than a category effect, right? Because there
yeah.
are difference in looking times coming from the fact that they're not familiar with the objects, rather than saying these two are in different categories, which is what they're trying to measure.
Yeah,
Am I making sense, or
yeah, makes sense, makes sense. Now I start to confuse.
Mm,
Mm. Yeah, I also spend some time in understanding this DLT, difference
mm,
looking time, and then the logic behind.
mm,
Okay, so maybe I'll lead this second question later, and then I can take more detailed look and we can discuss in qualia structure meeting. So
mm.
for first one, I don't know whether you have take a look at the qualia structure proposal.
Probably. Probably
Yes.
I should, because there's probably something on it. Yeah.
Okay, can I share my screen?
Yep.
Oh.
Go on.
So this is in Japanese. So I don't know. This is the similarity rating.
Mm-hmm.
And then we are going to find the mapping on in brain.
Mm.
And then we go back to brain and find the mapping of similarity rating. So that is the second object of the qualia structure. So that's why I think that's why they are trying to map or compare, for example, similarity rating and fMRI data. Did that explain a little bit? I'm not sure. Okay, I'm stop sharing the screen. No, wait, how can I do that?
We just there's a red button stop share, right?
But can I add some?
No?
comments?
I can just kick you out.
Yes, you can keep.
In this
Yeah, Yep. Yeah.
The slides we've been
Or
showed
is it in the
that
qualia structure?
yeah the second target goal to said they plan to measure the brain activity when we manipulate the qualia structure, and then they measure the qualia structure qualia structure when they manipulate the brain activity. There are two steps.
Yeah, actually,
to
not manipulate. It's finding the mapping of phenomenal data and the neural data. So maybe manipulating is the eventually goal, because manipulating human conscious.
I think the ping dropped out.
Business is
Oh.
Can you see that? Okay, so in this paper, they have we have data of humans or adults similarity rating by a hypothetical model, right? And then measure their brain activity. We have so they have we have data from adults of both. The first is neural data.
I think it's dropping out again.
And the second is similarity by eye movement. And in the introduction, they say that the similarity is correlate or related to brain activation pattern. So we can deduct or build a connection to this part.
Yeah. Wait, was it in the introduction? Oh my God, maybe I've not read it enough.
Yeah. Does it make sense to you?
Okay, so hypothetical model is correspond to the adult eye looking time data. Is this correct?
I think it's very laggy.
Similarity. So eventually both data are going to be this similarity rating from 0 to 1.
Can you repeat that, Liping? I think you dropped out for some while.
Oh, sorry. So for adult, we use hypothetical model, and then this hypothetical model the data type is also dissimilarity rating, and then this eye movement or glance time or gaze time is also will be converted into dissimilarity rating by the formula you post answer. So their data is they are all same data structure in metric. And then the dissimilarity is 0
Yep.
to 1. So
Yep.
essentially they are in same structure, same data.
Yep.
Yeah, I know. So my question is, I thought before comparing infant looking time dissimilarity data and fMRI data in adults, I thought they should compare the looking time data from adult and fMRI data from adult.
Okay.
But in my understanding, you said that the hypothetical model is really similar or corresponds to the adult looking time data. So they first compare these two, and then compare the infant looking time dissimilarity data and fMRI data. Is it correct?
Sorry, I didn't get very much. Can you explain it? Yeah.
To say something from experiment 2, I thought they should compare DLT from adult and fMRI data from adult. But you said before, in my understanding, hypothetical model is really match to the LTT data from adult. So they compare hypothetical model and infant LTD DLT data first, and then they compare the fMRI data and DLT data from infant. Is it correct?
I think
Yeah.
okay, Anson.
I think I think you mentioned the problem is that there's no link between adults dissimilarity rating or DLT or whatever, adult phenomenological data with adult fMRI data they've just introduced. The problem is that then I think, just in my opinion, I think they can't draw the conclusion about anything in experiment 2 because you don't have a positive control. Right? You expect that there are correlation between adult fMRI to adult phenomenological, only then can you infer anything about infants. But if you have not shown any correlation in the first place in adults, then you can't talk anything about infants because you have not shown that it is positive, right? It must be true. Right,
Yes.
so yeah, the second part was really confusing to me. That's right, yeah.
I agree with you. When I read this paper, I draw this graph and then I found they actually lack this correlation between adult similarity and then fMRI. So yes, that's a good question. I agree with you. And regarding the adult looking time, my thinking is that adult looking time may be not because why we are measuring looking time is that infant cannot speak, but adult can. So I don't think we need to measure adult looking time as a measure because looking time is a index of categorization or similarity we infer from infants and adult we can speak, right? We can speak.
It will be a good validation tool, I think. Just to say, well, in infants, we're using this paradigm. In adults, we are using a little bit more sophisticated and hopefully a little bit more close to what we want to try to measure in terms of dissimilarity. And it would be great to have a tie to say that actually in adults, we do see this correlation in DLT versus dissimilarity mating so that it supports the use of infants DLT as a measure for dissimilarity. But I don't know. I'm not an expert in infants research and all that stuff, but that, yeah. It'll be interesting to see, yeah.
You make sense. I think both of you are make sense. Yeah.
Can I ask a elementary question? So, looking time data is looking time data phenomenological data or behavior data? I think eye movement or looking time is behavior data, but you
yeah, I think
right.
you're right. Sorry. The term I use is not, I think it's not correct. You are right. It is behavior data.
Oh, okay.